Hey everyone, we're on a tight schedule and it's 9 o'clock, so I guess we're going to get started. Hello and welcome to the joint Spinach Tagwig 2024 conference. If this is not the conference you're here for, you should not be here then. But also congratulations on managing to get this far. This session is titled CTRB, Contributed Oral Presentations, a Potpourri of Presentations. This year's meeting was made possible in part by the generosity of our sponsors, especially our diamond sponsors. We are grateful to them and to the OK Congre for today's support in this conference. If you are attending virtually, please ask questions using the chat feature inside of Zoom. We will ask them to the presenters for you. And after the session, if you want to continue the conversation, feel free then to move it to the Oxford abstracts. That will save the conversations, unlike Zoom, which disappears. And a final reminder that by attending this conference, either virtually or in person, you have agreed to abide by the Code of Conduct. And now I would like to introduce our first speaker, presenting virtually via recording, Clara Baringo Fonseca, presenting Darwin Core Brazil, translating examples and open discussions.
Hi, everybody. I recorded the presentation. Can you know? Oh, I present directly here. Wait. Good morning, everyone. It is a pleasure to present at this conference, and I would like to thank the entire organizing team and all the participants. My name is Clara Baringo, and I am responsible for data mobilization at the Brazilian Biodiversity Information System, the GBIF node in Brazil. Today, and on behalf of the CBR team, Carlo, Kayla, and Johanna, I will be introducing an initiative called Darwin Core Brazil, a freely accessible GitHub platform where we translate the Darwin Core to engage the community and improve the organization and publication of data through platforms like CBR and GBIF. To provide some context, the Biodiversity Information System, launched in 2019, is a platform coordinated by the Ministry of Science and Technology that brings together occurrence and ecological data from across Brazil. Additionally, Brazil has implemented and customized the Living Atlas platform to manage its own taxonomy and to integrate and visualize occurrence data. Finally, we have been a voting member of GBIF since 2019. The use of Darwin Core standard for sharing biodiversity data and information has been increasing in Brazil. There is a growth in data publications on national and international platforms by institutions, foundations, programs, and research projects in biodiversity. However, due to the heterogeneity of data types, collections, and taxonomic groups, doubts persist about the adequate completion of each field in the dataset. Although the Darwin Core standard has resolved much of the issue, the open content for some terms complicates the search and comparison of data and information across different datasets. In order to facilitate the understanding of the Darwin Core term content, the Brazilian system has created the page Darwin Core Brazil with translation, best practices in using each term, as well as examples for the entire community. You can access to the Darwin Core Brazil page directly from the main page. Accessing the GitHub platform, you will find that the translation is almost identical to the official page. The main reasons for translating the Darwin Core are to improve data quality in data publications across the country and to engage the scientific community in discussions about the terms. Researchers and specialists in various fields can discuss terms within their areas of expertise, helping to define control vocabularies and best practices for filling each term. This approach facilitates the search and comparison of Darwin core terms, such as sampling protocol or sampling effort, among others. There are several initiatives in Brazil that are benefiting or will benefit from the implementation and translation of Darwin core. The first of these was the publication of a report assessing the quality of data from the Brazilian system. The Brazilian Institute of Geography and Statistics downloaded over 22 million records from the system, analyzed information regarding taxonomy, dates, and coordinates. The taxonomy groups analyzed include amphibians, arthropods, fungi, mammals, vascular plants, and reptiles from different sources, including project data, citizen science, and natural collections. Indicators on the quantity, age, and representativities of the records were mapped across the country. The second chart displays in dark green areas with higher number of records, while light green or white indicates location with few or no records. With the exception of birds, the proportion of records with complete information, including date, geographic coordinates, and species identification, is below 30%. The main limitations identified in the data were the absence of geographic coordinates and the lack of detailed species information identification. Despite these issues, the proportion of more complete records has been increasing in recent years. The results highlight how information can sometimes exist, but may not be well organized according to appropriate Darwin core terms. The translated Darwin core Brazil can help to improve the quality of records published in the system. Another example where Darwin core Brazil is important for the publication is important for the publication of data of environment recovery programs. The Fundão Dam collapsed in Mariana, Minas Gerais in 2015. Approximately 39.2 million cubic meters of tailings reached the river. As a result, was created the Renova Foundation. This foundation is responsible for the mobilization of the reparation of the damages caused by the collapse. This foundation brings together technicians and specialists from different areas of knowledge, dozens of environmental entities, and scientific knowledge from Brazil to do research. All this data is going to be published through national platforms. There are monitoring and data collection programs with various sampling protocols and measurements, where the correct publication of terms with controlled vocabularies is essential for data comparison and subsequent analysis on, and for that reason, Darwin core Brazil will be also important. Finally, discussing extensions and standards for socio-biodiversity data is also crucial. Recently, the GEF approved a project coordinated by the Ministry of Science and Technology in collaboration with the Ministry of Indigenous People and the Environmental Ministry, aimed at empowering indigenous peoples and local communities to manage biodiversity data and information. This initiative is a strategy to conserve their territories, safeguard traditional knowledge, and promote integrated biodiversity management. In this context, again, the Darwin core Brazil platform could be adapted to initiate discussions, to start discussions on standards and that could contribute to the project. So each term includes access to the official Darwin core, a Portuguese translation of the definition, and examples for each data type in Portuguese, along with an hyperlink to CPR. For written domains, we provide examples directly. For sampling protocols, we offer bibliographic references that describe the protocols as well as examples available on CBR. Experts can click on each term to start an issue. As you may know, issues help keeping discussion organized. For example, we show the discussion on the term behavior, where specialists in the field debate that the content and good practices for taxonomic groups. These discussions are reviewed by the CBR team and updated on the main page. Furthermore, the Brazilian community can organize to propose new terms. We also provide guidance in Portuguese on how to submit a term to TEDWG, and as we can see here, on suggestion and discussion of new terms and directly access to TEDWG website. Finally, additional translations are underway and being implemented. For instance, we are translating best practices and examples for completing metadata according to the Ecological Metadata Language Standard. This includes examples for publishing metadata on biological collections and metadata associated with research projects. So that concludes my presentation. Thank you very much for your attention. Please find our contact information below, and we are available for any questions or comments or ideas to integrate this platform together with TEDWG Quick Reference Guide on Darwin Court. Thank you very much.
Thank you, Clara. That was really fascinating for me to learn about because I do not know much about Darwin Core. So that was genuinely a lot of learning for me. We have a couple of minutes for questions. If anybody has questions, please either use the Zoom chat or come up to the microphone. Yes, yeah.
Hello, my name is Andrea Weeks. I was fascinated by your talk. I was particularly interested to see how you've identified gaps in your knowledge in terms of under-collected areas. And I'm wondering what feedback you have received from researchers who are perhaps inspired by these gaps and looking to fill them.
Thank you for the question. This was really good for our system because lots of people start to mobilizing more data. And yeah, so now we have like some researchers that want to, so there was like some programs like Amazonia plus 10 other projects financed by other countries that they now using this report in doing more like sampling, choosing better the sampling areas and the sampling and looking for better taxonomies for the groups there that have some gaps. And that's good also because researchers wanted to update their data for having more completeness of the data. So that was really good. And the idea of this report, it's available. I will put the link. Is to do it more frequently to compare how data is being better collected and better treated and qualified. Thank you.
Thank you.
Okay, we do have time for one more question if anybody has one. Okay, well, thank you again, Clara, for joining us today. I know it's very late where you are, so thank you very much for coming. Our next presenter is Deborah Drucker, presenting virtually as well on collecting, working towards plant pollinator interactions data, I can't read my own handwriting, I apologize. Interoperably and reuse lessons learned from the World Fair project.
Hello, my name is Deborah Drucker. I work as a data manager at Embrapa, the Brazilian Agricultural Research Corporation. And I'm here today to present the results from the case study on plant pollinator interactions data, one of the case studies of the World Fair project. The World Fair project was carried out from June 2022 to May 2024 and was an important partnership between CODATA and the Research Data Alliance, which are major organizations for global scientific data. Funded by the European Union through the Horizon program, the main objective was to advance the implementation of the FAIR principles in a range of disciplines and cross-disciplinary areas. I think at this point everyone knows what FAIR is, but in case I'm wrong, it stands for findable, accessible, interoperable, and reusable. We had 11 case studies, and the agricultural biodiversity addressed the challenges of advancing interoperability and mobilizing plant pollinator interaction data for reuse. GBIF coordinated a case study in biodiversity, and there were common challenges between our work packages. The agricultural biodiversity case study was proposed by IGAD, the Research Data Alliance community of practice dedicated to agricultural data. As a community of practice, IGAD encompasses several communities, and we realized there was a huge interest in plant pollinator interactions data worldwide. To name a few, the Biodiversity Information Standards Interest Group from TDWG on biologic interactions data, and the SURPASS project, a collaboration between several countries in South America and United Kingdom. We gathered collaborators from several organizations and proposed this case study, namely the Mais Botanic Garden in Belgium, the Kenyan Agricultural and Livestock Research Organization, the startup HiveTracks, and the RDA Association. We also had collaborators from in kind from several organizations worldwide, including the Brazilian Network on Plant Pollinator Interactions, REPIP. Our case study objective was to ensure fair data for understanding plant pollinator interactions at biologically relevant scales for crops. In the first year of the project, we conducted task 10.1, which we call discovery phase. In the second year of the project, we conducted 6 pilot studies to adopt the standards recommended in the discovery phase. So in a nutshell, in the discovery phase, we conducted a landscape analysis of the plant pollination data practice worldwide. We found that a huge amount of data is in supplementary files of research articles, and most of the ones we found are lacking fairness in many aspects: data formats, lack of semantic resources, lack of metadata. The diversity of approaches and the lack of appropriated vocabularies lead to confusion, information loss, and the need for complex data interpretation and transformation. And this is a challenge for researchers, data administrators, editorial boards, publishers, and scientific societies. This deliverable was published in Zenodo and contains the recommendations for the pilot phase. The 6 pilot studies in the pilot phase were observations of plant pollinator interactions in the Pampean region of Argentina, the Brazilian Plant Pollinator Interactions Network, HEBIP, plant pollinator data collection by CARO in the African continent, USDA Agricultural Research Service and Natural Resources Conservation Service, data collected through the HiveTrax application by beekeepers, and the plant pollinator interactions in wild ecosystem in Colombia. In the pilot phase, we adopted a data standardization strategy based on the following initiatives. The data schema and plant pollinator interactions vocabulary documented in this paper from José Salim and collaborators from 2022. The ecological metadata language, the biotic interactions platform, which aims to make existing species interactions datasets easier to find, access, and reuse using open source software. And together with GBIF, in 2 of the pilots, we explored the unified data model for biotic interactions and tested the beta version of the integrated publishing toolkit based on events. In September last year, we promoted a webinar together with GBIF, and if you're interested in this particular topic, I can send you the link to the recording. Our work with the pilot studies applying the data standardization strategy allowed us to develop best practice and guidelines, recommendations, cost of adoptions estimates, and a fair data assessment tool. You will find more details about all of that in deliverables 10.2 and 10.3. Globi developed a dedicated webpage to the pilot studies. Some of them are totally open data, and you can explore everything, including the data review reports. We also developed a cookbook to be used by people interested in adopting our strategy and making the data fair. We developed a tutorial to standardize a plant pollinator dataset with OpenRefine. A fair assessment tool. All the links to the outputs of our work are available at the World Fair webpage. Moving from diverse approaches and siloed initiatives to widely available fair plant pollination interactions data for scientists and decision makers will enable the development of integrative studies that enhance our understanding of species biology, behavior, ecology, phenology, and evolution. I hope you find this work useful, and here are my contact if you would like to discuss anything further. Thank you very much for your attention.
Thank you, Deborah, for sharing that. That's really cool. Again, we have a few minutes for questions if anybody has them. Deborah, I don't think anybody has any questions, but I found your conversation really fascinating. I think that the idea that scientists are putting all of the available data in annexes and not necessarily providing keys to them is kind of counterintuitive to being a scientist and providing data. And so I think what you're doing is really important to trying to fix that. So thank you very much. If anybody has any other questions, you can always put them and continue the conversation in Oxford Abstracts. So.
If I may, I just wanted to acknowledge that Esteban is also here in the Zoom call, and he was one of the members of the Colombian pilot. So this was a work from a large group of people from several countries, and I think that was the special thing about this project, was having so many people from different experiences and different data. And it was great working with everyone. Thank you all for your attention.
Thank you. I do think we have a couple of minutes until the next presentation, and we want to make certain that we stay on schedule and don't get ahead of ourselves, so hang tight for just a few minutes. Okay, our next speaker is Carlos Garcia Robledo, who I believe is yes, okay, he is here in live person, yay. And his talk is entitled From Data Points to Data Swarms, Preserving Specimens and Their Interactions in Tropical Food Webs.
Hello, everybody. This is good? Yep. Excellent. So I'm Carlos, and I am an associate professor at the University of Connecticut in the US, and I am an evolutionary biologist working on climate change and multiple species, but I think that I'm also the worst nightmare in the future for many of you guys working with collections, because I work with so many collections, so many specimens, interactions, and this talk is motivated because when I saw some of my colleagues retiring, they had these gigantic fridges full of specimens, and they just left. And then collection managers have to deal with that. So I'm trying to be a good citizen. All right, but this is also my first time in SPNHC. So I need to talk a little bit about myself. So I was born here in Colombia, in a city called Bogota, and this is more or less where I have been working for all these projects. So I am an associate professor up there in Connecticut, but also we have been working in mountains, in Los Tuxtlas in Mexico, in multiple sites in Costa Rica, where we do most of my research, and the Andes and the Amazon, and also the Peruvian Amazon. So the main goal of my projects are to understand how climate change will affect interactions, and we work in multiple elevational gradients in the Americas. So imagine that, we have all these different interactions happening there, but we have a core system that is called the Zingiberelles. This is a group of plants called the banana-like plants. I'm sure that if you have been in the tropics, you have seen this. They have the heliconias, the bananas, the gingers, and so on. And these are all the interactions we work with. So here we have some examples. We work with the chemistry of the plants. We work with the gut bacteria in the beetles that they use to digest their different host plants. I'm not going to talk about that now. But look at this, all these universe of interactions. So a few of them are, here we have the beetles. So these are herbivores that feed on the Zingiberelles. And this is one of the oldest and most conservative interactions between plants and insects. It is supposed to be one of the oldest, 60 million years old. So basically, when these beetles were feeding on Zingiberelles, the dinosaurs were around. So super, super old. And it is becoming one of those model systems to understand ecology and evolution between plants and insects. So roll leaf beetles live inside the rolled leaves. These are young leaves that are rolled. They don't roll the leaves. And what they do is that if you insert a young leaf of a gingiberelle in the neotropics, this is what you're gonna find, a bunch of different species of beetles feeding inside that roll. So when these leaves mature and unfurl, these beetles have to fly and they have to find a new roll leaf to start the life cycle, all right? Okay, but these beetles are super diverse. So imagine that I'm working on these different elevational gradients in the different mountains. And then one of the big challenges of my project at the beginning was that to understand interactions and climate change in this system, you have to know which beetle species are present at each different elevation. So what is a species, right? Then you have to know which beetle species is feeding on these different host plants, but also how these diets of these beetles change with elevation and latitude. So for this is kind of like old news now, but when I developed this method, it was kind of like a cool thing to do. So, and everybody kind of like based their method now on this original project that we did with these beetles. So what we do to understand diets is use short sequences of DNA that we call DNA barcodes. So what we did was to obtain DNA barcodes for all the potential host plants of these insects along these elevational gradients. Then after that, we developed methods where you can extract DNA from the gut contents of the insects, and then you can start matching beetles with their diets, right? So here we have one of those data swarms I'm working on. But when the beetles are flying to these novel host plants, they don't hitchhike alone. They have this group of mites that you see here, and you can see here too, right? And this is a super diverse group of mites that are phoretic. So they hitchhike on the beetles, and they move from one roll leaf to another one, and they depend on the beetles as little planes to move to the different host plants. So super diverse, right? We have new families still undescribed for this group. We have mites that can be, some species can be symbiotic, so they feed on a fungi that grow on the elytra of the beetles. And if the mites are not there, the virus will die because the fungus can kill them. We have some mites that have these modified legs, and they swim in the little layer of water in the lower leaves. They are filter feeders, and they have these little filters to feed on little organisms. We have predators of nematodes, or we have predators of other mites. So a universe of interaction that is crazy. So for some of the projects, we need to understand the interaction between plants, associated insect herbivores, and also associated mites hitchhiking on the virus. And this is for some projects where we're trying to understand the process of extinction and co-extinction in complex trophic webs. So look at this. We have, for one beetle species, we can have something like 10 mite species. So we have one order of magnitude of increase in diversity for this group. So imagine that, so many organisms here. So what we did for this is that we have, in addition, for every single beetle species that we have in our data set, we have a DNA barcode associated with the specimens. But also we have thousands of DNA barcodes for all the different mites. So we know the mite that is hitchhiking on each one of the different beetles to move to different host plants. Here we have another interaction. We have pollinators. So this group of plants are mainly pollinated by hummingbirds, but also by euglossine bees. So these are these beautiful green, colorful bees that you see in the tropics. And here we have an example of the kind of data that we have for one location. This is the La Selva Biological Station, that is a tropical rainforest in Costa Rica. At the bottom, you have the different hummingbird species in this location, but then the rows represent the plant species, and then the dots represent who is what, right? The typical bipartite network of interactions for this elevation, for this site. And imagine if you have this for multiple elevational gradients in different locations. So data swarms. And then here we have the last interaction I'm going to be talking about today. So there is a hummingbird at La Selva Biological Station feeding on the plant, and everybody can see what's happening in the beak there. You see those little dots like moving around. Those are more mites, but these are mites that are totally different from the mites that are hitchhiking on the beetles. These are called the flower mites. This is a group of neotropical mites that live in the flowers. They feed on nectar inside the flowers, and the only way they can hitchhike from one plant to another one is by really quickly get on the hummingbird beaks and then be able to move to the next plant, right? Okay, so here we have all these different interactions. Here are the mites, look. So for the mites, we also have another data swarm. We have at the bottom the different flower mite species that are at this place called La Selva Biological Station in Costa Rica. And here we have the different host plants. So this is another group of mites that we work with. So what you can see in the video is the hummingbird visiting. The flower has a little window. And then we have this high-speed camera that will, look at that. They cling on the beak there. Whoop. Super cool. And one thing that we just discovered is that these mites are using electricity, and they are able to detect electricity from the hummingbirds to hitchhike on different hummingbirds. Super cool. So stay tuned for that. So the challenge. The challenge is that species concept is a hypothesis, but it is my hypothesis, right? Then how can I actually make sure that people understand what is my hypothesis for what is a species with all this universe of interactions, so my studies can be replicable? And how can other researchers test my hypothesis? And they can just do my studies again. Okay, so each interaction must be represented by a museum voucher. And that's something that I'm trying to do with everything. So we have museum vouchers in the locality, in the museums in the locality where we're doing the work, but also at University of Connecticut. And this is kind of like an example of how we kind of like process the specimens from the specimen to the DNA barcodes that we extract. Then we send this to gene sifter and so on. But well, the thing is like this looks super complicated. And then at the end, all those data stay eventually in my computer. And then what you have is basically a DNA barcode that is published in bold, right? So there's a lot of information that is lost there. When I retire, that's not gonna be there. But then one solution that I had since I was a postdoc was that I started including in the Encyclopedia of Life one page for each single species that is described. So imagine that for the mites, it is impossible because most species of mites, we don't even know the genus, right? But for the species that we actually have sort of like a good taxonomy, we can do that. And for the interactions, what I started to do is that we started including in Darwin Core machine tags in EOL too, but this is super challenging too, right? Because we have, and actually this data set for the beetles and the plants was one of the models used by Darwin Core to actually include interactions in these databases. So that was super cool. And then over the years, these data points have been populated by data swarms from other sources. For example, I don't know, iNaturalist or so on. It goes there. So there is a place where some of the information that is hidden in my computer will be placed for other people to be able to use. And all of these specimens and interactions, what I'm doing is that we have DNF codes for everything, and we have specimens. So, I mean, I hope that people can repeat that. But then there is a big challenge, right? Because I am an evolutionary ecologist working on climate change, but at the same time, what am I gonna do with all these species? Who's gonna describe them? So at some point, what I had to do was, because I think this is important, I became the taxonomist for some of these groups. So it is a lot of work, because I have to combine writing the papers about the concepts that I'm working on in the laboratory climate change, models to predict changes in populations and communities of these organisms, and also warning about the insect apocalypse, right, that we're talking about just a few years. So we have associated publications to these publications that are taxonomical. So people can actually go hunt for that information there. Okay, so some challenges including interactions in biological collections are, there are few incentives for ecological research to carefully preserve vouchers. It is challenging to get funding to preserve specimens associated with ecological research. And also it is a lot of work. It is a lot of work. So what inspired me to curate interactions together with my specimens? So I had my first postdoc at Smithsonian Institution, and I was a dual fellow. I was a dual fellow in ecology and entomology. And I was in an institution where it was really important to actually have accurate taxonomy for the work that you're doing. So that was really good. And also I had funding. So in addition to have my fellowship, the Encyclopedia of Life had these amazing fellowships, thanks to E.O. Wilson, that gave you money to actually sit and work with the taxonomy of your organisms. So I took advantage of that. So in conclusion, ecologists should, well, if they can, you're right, because it's what they want to do, increase the replicability of their experiments by using biological collections. For this, we need incentives. So for example, additional funding, something, right? If not, we won't do it. We need to develop plans early in your careers for specimen management. I've been working on this group of organisms for like 20 years since I was a grad student, and figure out how to do this curation, but it has to be in a way that doesn't suck all your time into doing this, because I have another job, right? And ecologists need to interact more with collection experts, and that's why I'm here. Thank you very much.
Thank you, Carlos. Unfortunately, we don't have any time for questions, but we do have a few minutes at the end of this session for more of a round robin of questions. So Carlos, if you're able to stick around, maybe you can ask them then. Our next talk is by Destiny Gonzalez and is titled Historic Collections Can Save Lives, Integrating Natural History Specimens into Pandemic Forecasting.
Sorry. Okay, so many museums have worked on integrating themselves better into One Health initiatives, but many more should consider building stronger ties with these strategies. One Health is an approach that recognizes the interconnectedness of human, animal, and environmental health, yet there's historically been less emphasis on how local ecology is impacted. 75% of all emerging diseases originate in wildlife. 60% of infectious diseases in humans are spread from animals. This can be due to a range of activities, such as rapid urbanization, leading to habitat loss, wet markets where live animals are kept and sold, as well as the consumption of bushmeat. So how can museums fit into One Health initiatives? We can begin by providing accessible data for research through the vouchering of host specimens. If we build collections correctly, we could have a huge array of sampling from all taxonomies. I believe that with time and dedication, we could do our best to be proactive with our collections. The Museum of Southwestern Biology, or MSB, is a research collection and biorepository associated with the University of New Mexico in Albuquerque. It contains 9 different divisions, 2 of those being the Division of Mammals and the Division of Genomic Resources. Within the Division of Mammals, we maintain a large collection with over 350,000 specimens. The collection has global spatial coverage, deep temporal coverage beginning in 1880, and a large taxonomic coverage as well. As the second largest mammal collection in the world, we've been able to grow our collections by an estimated 250,000 specimens over the past 20 years, largely through deep and ongoing collaboration with federal agencies such as the CDC or the U.S. Fish and Wildlife Service. Many of these collaborations were through emerging pathogen projects. This map shows the MSB collection spatial holdings. We have a large focus in the US and South America, but we've been able to branch out to a lot of different countries and collect in these as well. MSB is engaged in several emerging pathogen projects, but perhaps the earliest and best known example of this is with Sonombre hantavirus. MSB just so happened to be brought into the project and quickly became part of the discovery infrastructure. I'll briefly go over this pathogen and how it's an example of a reactive response to emerging zoonosis and why we're successful in becoming a part of that discovery infrastructure. So in 1993, an outbreak of a then unknown virus led to the death of young, healthy people in the Four Corners region, including the states of New Mexico, Colorado, Arizona, and Utah. In May of 1993, a young Navajo woman came down with a sudden respiratory illness that puzzled doctors. She quickly passed from this illness, leaving her family behind. On the way to her funeral, her fiancé quickly came down with similar symptoms: shortness of breath, coughing, and a fluid buildup in the lungs. Likewise, he died soon after arrival at the hospital. This prompted local health authorities to look deeper into these mystery deaths. During the first year of this outbreak, it disproportionately affected Native American communities in the region and had a 50% mortality rate, killing 13 people that year. Once local health authorities were called in, this soon became a national issue. This prompted a unique transdisciplinary approach to action. The CDC, Indian Health Service, Museum of Southwestern Biology, National Institutes for Health, and the Department of Health for the affected states all worked together to find the cause for this illness. MSB has maintained tissue samples archived since 1979, and were able to provide these for the research that proved that this illness was not new. Researchers identified Paramyces maniculatus, or the eastern deer mouse, as the reservoir host for this virus. Sonombre was the first hantavirus pulmonary syndrome discovered to affect humans. Before this, only one hantavirus in Maryland had been discovered, and risk to humans was relatively quite low. There have been reemergences of Sonombre hantavirus since the 1993 outbreak and is therefore still considered an emerging pathogen. Between 1993 and 2020, there were 833 reported cases, although the CDC released a study like 2 weeks ago that there have been inconsistent testing standards and there may be more or less cases than we know. Now there's a mortality rate of 35%, which is higher than old world hantaviruses, with mortality rates ranging from 1 to 15, but lower than the Andean hantavirus. Today, there are currently 1,004 MSB specimens cataloged into Arctos that Sonombre was detected in. Hantaviruses were previously classified as roboviruses, meaning that these pathogens are sometimes rodent-borne, being contracted through rodent excreta like urine, feces, and saliva. Now we know that this is old news. Since the initial 1993 outbreak, researchers have discovered hantaviruses in shrews, bats, and moles, all non-rodent reservoir hosts. Using metadata from collections, we've been able to better forecast where these hantaviruses may emerge next. In the MSB mammals collection, there are 18 viral symbiotypes, meaning the one type specimen in which a virus was discovered. Symbiotypes originally meant for parasites, but we tweaked a little to include viruses. These include types for Sonombre hantavirus, as well as Chocolo hantavirus, which became prevalent in Panama. Many of these viral symbiotypes are from collecting events, but others were described from specimens that had been in the collection for decades. We have a vital resource that only museum collections can have, and the only chance to be able to look back in time at these pathogens. Here is a brief chart of mammal specimens from different global emerging pathogen projects that MSB has taken a part in. In total, there are over 100,000 specimens associated with these projects sampled from all over the world, with many of these projects being long-term. We were able to grow our collections tremendously through this long-term engagement. So Sonombre hantavirus response was reactive. Researchers only found the source of that virus after it had already emerged and caused the death of many. With focus on building collections, we could detect viruses before they emerge and affect human populations again. So why should we focus on building our biorepositories when it comes to pathogen strategies? Large and natural history collections should always be growing. Our planet is changing now. With these changes, we'll likely see more zoonotic spillovers occur, and we can find ways to forecast these. Many collections can shift towards collecting samples from biological communities, not just focusing on one species, but instead sampling the symbiotic dynamics. Biorepositories can also act as a space for different agents and stakeholders to come together. The University of New Mexico's Project ECHO supports MEPA, or Museums and Emerging Pathogens in the Americas, which is a collaborative program across many national and international collections to come together, along with others who may be interested in the topic, and have conversations about ways that museums can better integrate into One Health. With collaborations like this also comes the need to build capacity and infrastructure, particularly in middle and low-income countries, for biorepository-based pathogen identification, surveillance, and mitigation. The United States has many museums that have established themselves in pathogen research, but support is crucial for other countries to be able to provide the space to maintain samples from tissues to skins. Without sampling and support of these countries, we could be missing out on invaluable data. Many biology museums also utilize large databases to manage their collections, provide and track metadata and samples. An example of this is Arctos Collection Management, which MSB uses, which stores almost 5.5 million records from 243 collections. Vast data stored on Arctos, including the usual important pieces like locality, sex, measurements, and whatnot, but can also include serological data and then genetic sequences available through GenBank, relationships with other specimens in the museum's divisions like parasites, and citations for an individual specimen. Arctos is meant to be a collaborative and accessible management solution that ensures high-quality data that can be used in future pathogen-related studies, but a huge issue comes through in its usability. It is a very powerful tool, but only for those who know how to use it. If I can learn it, it is possible, but we have to build front-facing access to this data for non-museum folks. Altogether, natural history collections hold vast data. MSB alone, like I said earlier, maintains over 350,000 specimens and over 650,000 cryopreserved tissue samples. If we can find ways to create more collaborative spaces for museums, imagine what kind of research all of our data we collect could contribute to. The data is only useful if we emphasize and encourage host pathogen researchers to deposit specimens into collections for future studies, or the practice of vouchering. Thompson et al. Says it best. Vouchering specimens and associated samples provide both repeatability and extension to host pathogen studies, and using them mobilizes a large workforce to assist in pandemic preparedness. Vouchering is crucial to be able to look on to future pathogens that will eventually take place. So in order for our data to reach its maximum potential, we must begin sampling holistically. Metadata is garnered through holistic sampling, collecting skin, skeletons, tissue samples, as well as ecto and endoparasites, blood samples, locality and habitat data, and more. This also entails collaborating with a broad suite of expertise in the field. I am a mammologist by training, but often work with parasitologists, virologists, and others in the field in order to boost our collecting potential. We maximize the scientific value of a collecting event when we collect with intent, understanding exactly what we're taking, why we're taking it, and what can be done with it in the future. This data may be used for any number of scientific concepts and issues, not just emerging pathogens. And when it comes to infrastructure, many of us know that museums today store tissue samples in -20-degree freezers or in ethanol, but as many of us also know, that DNA degrades in these conditions over time. So what's the best plan of action for preserving tissues so that they can be utilized for future research? MSB was able to upgrade to vapor phase nitrogen tanks beginning in 2016, thanks to NSF funding, as well as institutional funding through the University of New Mexico. And you can do it too. Building and upgrading our own personal infrastructure can allow for museums to truly become a space for pathogen monitoring. If we build our collections with this material in mind, we have the tools to provide for research that allows us to be proactive when dealing with the next pathogen that emerges. This is only possible if we are proactive now about how we move forward with growing our museums. Thank you.
Thank you. We do have time for questions.
Hi, good morning. Just a quick question about suitability of historic specimens in collections. Are you saying you detected hantavirus in old mouse specimens, but is just a single preserved skin enough, or is this talking about having a tissue sample cryogenically preserved? You know, how historic a collection could you go back and sample from?
Yeah, so like I said, we had samples dating back from 1979. A lot of these were stored in freezers or in ethanol, so we were able to pull data from those. But obviously, it's not optimal to store data or DNA in that. I think tissue samples are the best way of pulling information for viruses. Skin clips are not always the best, and yeah.
Thank you for the presentation. So I really haven't, I'm a botanist. I don't think about One Health very much, but I am aware of it. But I am curious, at least within the United States, are you aware of NIH, National Institutes of Health, funding voucher specimens? Like, supporting the museums that are providing these data?
Yeah, so MSB has been able to get a range of funding from different places. So because NIH was one of the agencies that we worked with in 1993, we've been able to get funding through them, but I'm not sure if they have broader funding available. So, yeah. A couple more questions. Anybody else? Thank you.
Okay, we are a few minutes, okay. Yeah, we are a little bit ahead of schedule, so take another brief break and then we'll have our next talk, okay? Okay, our next talk is by Natalie Masick. Marine Mammal Recovery is associate, this is the wrong one. Yeah, that's the wrong one, guys. It's the wrong presentation. We're fine. There we go. Okay. Yeah, metalinastic, everybody. Marine mammal recovery is associated with the resurgence of a nematode parasite.
Thank you. Hi, I'm Natalie. I'm a postdoc at the Yale Peabody Museum, but this is research that I did as part of my PhD in Chelsea Wood's lab at the University of Washington. And I know this title doesn't have anything to do with collections, so I appreciate you being here anyway. We'll get into why it's relevant. But first, we're going to mentally travel across the Pacific to the Puget Sound in Washington State. Every year, whales and marine mammals draw in thousands of visitors to this area, so much so that they have become an icon of the Pacific Northwest. But marine mammals were not always as loved as they are today. Until the 1970s, populations were driven down by population control, whaling, and live capture for aquariums. And the result was a Puget Sound that had way fewer top predators and marine mammals than there were historically. And then more recently, our relationship with marine mammals has shifted. In the 1970s, the Marine Mammal Protection Act was passed, and this protected all marine mammals in US waters. As a result, populations were given the opportunity to recover, and we saw increases in harbor seals, stellar sea lions, and humpback whales in this region. I am a marine mammal parasitologist, and so I was wondering if there's been this increase in marine mammals in the region, if there's been a corresponding increase in their parasites as well. And the parasite that I was the most interested in was the family Anisakkidae. We call this Anisakkids for short. These are one of the most common marine mammal parasites. They take up residence in the intestinal tract of marine mammals, and they are parasitic nematodes. They have multi-host life cycles, which we'll get into in just a second. And there are three main genuses of this family that are relevant to this talk. Anisakis species that use cetaceans or whales and dolphins as their definitive hosts, Pseudoterranova that use pinnipeds or seals and sea lions, and Contrasecum that use pinnipeds and seabirds as their final hosts. So how do these marine mammals get infected? Anisakkids all have the same general life cycle. So they start as eggs that are released into an ocean through a marine mammal's poop. They then get consumed by a small copepod, and they develop into their next larval stage. And then they keep doing that up the food web until they return to their definitive host, that marine mammal, where they grow to their adult stage, reproduce, and start that cycle over again. So depending on which host the nematodes are in, they infect different areas. In intermediate fish hosts, they often insist in the muscle, like this. And in the marine mammal themselves, they inhabit the GI tract, and they can cause inflammation and ulceration of the stomach lining, and also reach huge quantities, like in this minke whale. So there is evidence that in other regions where we see marine mammals increasing, there's been a corresponding increase in their parasites. This happened in the Baltic Sea with gray seals. Up until the early 2000s, there were very few gray seals in this region, and there were very few corresponding anisakkids in Baltic cod. And then these seals, whose populations had been controlled, were allowed to recolonize the area, and their population increased, nearly quadrupled over the course of 13 years. And we saw this huge increase in these parasites in Baltic cod. Over 50% of large cod had these Pseudoterranova worms, and 100% of them had Contrasecum. So is something similar happening in Puget Sound with increasing marine mammal abundances? To see how marine mammal parasites have changed over time, like in the Baltic Sea study, we chose to look at fish that marine mammals eat rather than the marine mammals themselves, because as you probably know, marine mammals take up a lot of room in collections, so historic marine mammal samples are kind of hard to come by. And because published data on parasite infections in fish are pretty scarce, we had to look inside old fish. And we found a lot of old fish available in the fish collection of the Burke Museum at the University of Washington. So how did we know we could find parasites in these fish? Well, two things had to happen before we could do this study. The first was that Ingrid Howard and colleagues worked to find out if you could detect parasite change in museum fish. They looked at the parasite Clavonema mariae, which is a really big nematode that insists in the skin of flatfish. You might have seen this. It forms dark red blisters. And this parasite was ideal for this study because it's easily visible on both fresh and preserved fish. So you can assess if a fish is infected without dissecting any museum specimens. There are also historical government data on parasite prevalence in English sole. So we could compare those historical data to data that we collected from the museum fish. So the authors did this, and they found a significant correlation between the historical data and the parasite counts from museum specimens. Cool. Okay, the second thing that had to happen was Evan Fiorenza and his colleagues assessed if parasites were as detectable in fluid preserved fish as they are in fresh fish. So the authors collected a bunch of fish from Puget Sound, and they froze some of them and fluid fixed the others. And then they compared the parasite prevalences between the two, and they found no significant difference in the detectability of parasites. So with these two studies, we know that we can detect similar trends to those logged by people that were looking for parasites in fish. And we knew that we could detect as many parasites in fixed fish as we could in fresh fish. So we knew that we could dissect museum fish and find changes in parasite abundance over time. So we worked with the Burke Museum's fish collection, and we selected fish that were really well represented both in the collection and across time from Puget Sound. Our aim was to dissect 10 fish species per decade, and most of these specimens were collected from 1920 to 2018. So we dissected these 5 species of fish and selected them because they are major prey items for marine mammals in this region. And because these were museum specimens, we had to be really careful about how we handled them. So we did a small incision along the belly of the fish and carefully removed all of the organs, looked through them for parasites, identified the parasites morphologically to the genus level, and then returned the organs to a smaller jar in with the fish, so the entire specimen is still complete. So with these data, I had 2 main questions. First, what are anisakids doing over time in Puget Sound? And second, what is driving this change? So 2 big things that have changed in Puget Sound over this period are marine mammal abundance, like we've been talking about, and also climate change. So sea surface temperature has increased in this region, and we expect both of these could contribute to changes in parasite abundance. So for our analysis, we aggregated all of the parasite data across host species and accounted for host species in the model. We ran state space models that reconstructed the population estimate for each of the parasite species to determine how they changed over time alone, and then with respect to these 2 drivers, so temperature and harbor seal abundance. We chose the best fit model to describe what was most likely changing or driving changes in parasite abundance in this region. So we ended up dissecting 448 fish and found over 2,000 anisakid nematodes. Most of these were Contrasecum, so we only ran the analysis on Contrasecum species. And as a reminder, Contrasecum are the ones that use pinnipeds and seabirds as their definitive hosts. So the data that we integrated into the model were harbor seal abundance data and temperature data, sea surface temperature data. Unfortunately, we only had data on what harbor seal abundances were doing from 1978 to 2013 that you can see here in yellow. And that's the best, longest temporal data on marine mammal abundance in this region, unfortunately. So you can see in the yellow that harbor seals increased and then dipped off and leveled off in the 2000s. Regional sea surface temperatures in green have been increasing steadily since the 1970s, and the average here is represented by that dashed line. And this is our estimated Contrasecum abundance based just on the state space model without the influence of any drivers. So you can see since the 1920s, we've seen a decline in Contrasecum species in Puget Sound with a tiny recent increase since about the mid-1990s. The best fitting model that we ran was one with harbor seal abundances included. So this is estimated Contrasecum abundance over time where their population growth rate is dependent on harbor seal abundance. And you can see, similar to the output without harbor seals, that we have this decline over time, followed by a more recent increase. And while we don't have harbor seal abundance estimates from before 1978, we do know that during this time, harbor seals were being hunted and culled until there were only about 1,700 seals left in the entire state of Washington. And then after the Marine Mammal Protection Act, their population increased and reached a stable point in the mid-1990s to 2000s. Current estimates for this population are around 11,000 seals. So while we see this marginal increase, it does suggest that Contra cecum is increasing with harbor seal hosts. And the reason that we could find this trend was through parasites hidden in museum specimens. So by using museum specimens as repositories for parasite data, we can better understand ecological change over time that would have otherwise gone unnoticed. This work was funded by the University of Washington and Oceans Initiative. I'd love to thank the folks at the Burke Museum that helped me with this study and were partners throughout the process, and the Yale Peabody Museum for helping me get here. If you're interested in a detailed account of how the Wood Lab works with collections to extract parasite data from collections, we wrote a how-to guide, which is available here. And I'll leave this up as I take any questions if I have time. Thank you. Oh, I won't leave it up.
Hi there, great talk.
Thanks.
I know it's outside the scope on one front, but is there any data on the bird populations to see if there are any increases or decreases that would associate as well? Thank
Great
you.
question. There are not consistent data sets of birds in this region. There is the Audubon Christmas bird count, which extends back, I believe, into the 70s, but it kind of varies depending on how interested people were in birds at the time. So with increases in birding, then we see increases in birds, which makes it not the most reliable data set. Thanks.
Great work. So I'm from the collection management side of things, and I open up specimens and I find parasites and I
Yeah.
remove them and give them to our appropriate department. As a parasitologist, though, would you prefer to see your little friends already in the specimens, or are you just as happy already having them in the invertebrate zoology or entomology or what have you department?
I'm happy for someone else to do the dissections. Yeah, I was talking to somebody about how long it takes to get woozy while dissecting museum fish. Yeah, as long as you document what part of the body the fish or the parasite was in, then it's super usable data for us. Yeah, thank you. Great. Any other questions? Thank you.
All right, thanks, everybody. Our last paper was unfortunately unable to attend today, but if you're still here, and if anybody has any questions for Carlos, this is an excellent time to ask them. So, and otherwise, have a good day.